Let's start off with the rationale behind this study. Skin disorders are often triggered by environmental factors, such as trauma and UV radiation, which leads to unwanted consequences. This is where Centella asiatica, or biboboc, steps into the picture. Previous studies have shown that it contains anti-inflammatory and antioxidative properties, in which is why it is used to treat various diseases. It is also widely used and highly accepted in the cosmetic world. However, in terms of its therapeutic use, the underlying mechanism is still unknown. Hence, the study aims to investigate the effects of Centella asiatica and its derivative on skin wound healing and UV-induced keratinocyte damage, as well as its underlying mechanism. First off, to investigate the wound healing effect, keratinocytes of the high-cat cell lines were used as a cell module. Once full confluence has been reached, the cells were scratched with a pipette tip to imitate wound formation. Then, it is treated with our extract and photographed in 24-hour intervals. If wound healing is successful, our extract contains wound healing compounds, whereas if the wounds remain open, the extract does not contain effective compounds that promote wound healing. Results show that only aqueous extract exhibit wound healing properties, and upon further analysis, we identified that there are two bioactive compounds, Madecassoside acid and Asiaticoside A. Up next, we utilize the pure compounds in varying concentration. The results for Madecassoside acid show that it enhances wound healing in time and concentration-dependent manners, with the optimum concentration being at 50 micromolars. The same can be said with Asiaticoside A. However, this alone, we cannot identify whether the underlying mechanism is through proliferation or migration. So first, we test for proliferation. To determine this, we utilize the BRDU proliferation assay. BRDU competitively binds to adenine acid-is thymidine analog and allows the attachment of primary and secondary antibodies, as well as an enzyme that allows the conversion of a substrate into a product where color change could be observed and the OD could be measured. The results show that both of these compounds dose-dependently increase the rate of keratinocyte proliferation. Yet this alone cannot establish whether the proliferative properties is what enhances wound healing. Therefore, we treated ourselves with mitomycin C and our bioactive compound. Mitomycin C binds to DNA, which suppresses DNA replication, hence eliminates cell proliferation. In these conditions, if wound healing occurs, we can say that the underlying mechanism is through migration. Whereas if wound healing is unsuccessful, it is suggestive of proliferation. In the Madecassoside acid and mitomycin C treated groups, we can see that wound healing still occurred, therefore signifying that the underlying mechanism is through migration. The same can be said when the cells were treated with Asiaticoside A. To investigate the underlying mechanisms involved in keratinocyte migration in wound healing, our cells were treated with various inhibitors that are commonly known to be involved in the wound healing process. If wound healing occurs, we can say that our compound does not utilize the signaling pathway of the respective inhibitors, whereas if wounds remain open, we can say that our inhibitor suppresses the effect of our compound, therefore indicates that it utilizes the signaling pathway. The results show that Madecassoside acid does not utilize the SIRS1 pathway, but instead utilizes the calcium, AMPK, ERK, and mTOR pathways instead. The same could be said with Asiaticoside A. Now we have established that both of these bioactive compounds can promote and accelerate the rate of wound healing. However, it does not utilize its proliferative properties. Therefore, we wanted to say whether there are other mechanisms that, other things that it utilizes the proliferation to protect against UV-induced keratinocyte damages. So here we assess this by using the MTT assay. Mitochondrial reductase in living cells help catalyze the reaction that converts MTT into pharmazan, which causes a change in color. So our high-cat cells were UV'd and then treated with our compounds, where the OD could be measured. Results show that both of these compounds increase cell viability in concentration-dependent manners. To visualize this further, we conducted the death cell assay where we stained our UV and treated cells with lithium homodimer and Hertz. In normal cells, the nucleus was stained blue as lithium homodimer cannot pass through the stable membrane due to its negative charge. In apoptotic cells where the cell membrane is partially dissociated, the nucleus will stain orange-blue as lithium homodimer can partially diffuse through. And for necrotic cells, the nucleus will stain red as lithium homodimer has a greater affinity to the positively charged nucleus. The figures show that madecassosic acid provides protection against UV-induced damages as it decreases apoptotic and necrotic cells, shown in this figure. The same can be said with asiaticoside A. To identify the underlying mechanisms involved in its protection against UV-induced damages, we did the MTT assay and co-treated with various inhibitors. It is seen that the underlying mechanisms involve the calcium, AMPK, mTOR, and ERK, as properties were completely abolished with the co-treatment of these inhibitors. The same could be said with asiaticoside A. Upon our Western blot results, we found out that calcium, AMPK, and mTOR increases ERK phosphorylation. To conclude this study, we found out that asiaticoside A and madecassosic acid increases intracellular calcium, which in turn increases AMPK, mTOR, and ERK activity, which leads to the enhancement of wound healing where migration and protection against UV-induced damages via proliferation. We have now completed the cell module in high-cat keratinocyte cells. However, in the future, we can incorporate the mouse model where, if successful, and move on to the general population. The benefits of this study is that it can lead to the successful development of inexpensive and effective treatments against skin diseases and from Thai herbs and promote agricultural products as therapeutic options. Now, lastly, I would like to thank my advisor, Assistant Professor Pawin Pongkopsakul, as well as Chulabhorn Royal Academy for their continuous support. And thank you so much for your time and attention.
Thank you very much for another valuable presentation. Does any judges need further clarification?
I'm wondering whether you have looked to see if there's some common pathway that share or control AMPK, mTOR, and ERK.
The common pathways. So upon literature review, we found that this is a common pathway used in wound healing already. So we were just here to confirm whether this could be the case. But I think the reason why both of these compounds also exhibit and utilize the same pathways is because of a common part of its structure. As for asiaticoside A, I'll show you one second. One second.
So there are 3-4 different pathways, and then
Yes.
your herbs act on all of them independently.
So because we didn't actually conduct it on more inhibitors, as you know, multiple signaling pathways can be involved. However, because of the literature review, we limited to just the few that were commonly involved. However, I think that for future perspective, I think it's also important to explore others because, as you know, there could be more than one, the signaling pathway that we discovered, there could actually be more. So I think we can move on to that after as a future project as well. Yes.
So have you purified this compound by yourself?
No, actually, so we didn't have to purify the compound by itself because this was bought commercially as the compounds can be purchased by a company. Yes, but because before we did the extract with just the extract from Centella asiatica and we identified it, we ensured that we utilized the same purified compounds from the commercially bought ones.
Okay, but you mentioned that it's aqueous phase of the...
Yes, so because it was bought separately afterwards, so after we found out about the pure compounds, we bought from the company the actual pure compound without the extract.
Oh, okay.
Yes. Thank you. Thank you for your questions.